Hi, I'm Lorielle Dietze, a PhD student in Dr. Tomas Hayek's lab in the Department of Psychiatry at Dalhousie University. I'm going to be talking about our research summarizing the associations between obesity and white matter. Obesity is becoming increasingly common worldwide, and in Canada alone, 63.1% of the population are overweight or obese. Obesity comes with various health risks, including cardiovascular disease, hypertension, and type 2 diabetes, among others. However, lesser known are the effects of obesity on the brain. More recently, there have been research investigating the brain changes seen in gray matter, where there are widespread alterations. A meta-analysis on obesity and gray matter again showed diffuse brain changes. Much less known are the alterations to white matter and obesity, where only one restrictive meta-analysis showed localized changes to the genu of the corpus callosum. Our meta-analysis investigated fractional anisotropy, a diffusion tensor imaging measure that looks at white matter microstructure from the diffusion of water along axon bundles in the brain. The water movement can be isotropic, diffusing in all directions, indicative of neurodegenerative disease, or anisotropic, where the water flows along the axon bundles, which is what we see in healthy brains. We searched the literature for articles containing obesity and white matter or anisotropy, screen these articles based on title, abstract, added articles from the previous restrictive meta-analysis not found in the search results, and assess the articles for eligibility. We were then able to split these into two different meta-analyses, a spatial meta-analysis and a region of interest meta-analysis. To answer the question, where are the brain changes, we used a voxel-based spatial meta-analysis, including 5,237 individuals across 30 articles, where we extracted MNI coordinates from the studies examining the association between obesity and fractional anisotropy. These peak brain coordinates from the studies were then combined using the SDM method, which calculates the probability of spatial overlap of these peaks. Our results suggest that obesity was associated with lower fractional anisotropy and specific localized white matter tracts. And these are including the genu and splenium of the corpus callosum, the middle cerebellar peduncles, the right cortical spinal projections, and anterior thalamic radiation. The other question we needed to answer was how large are these brain changes, or how large are the differences we are seeing with obesity? We used an effect size region of interest meta-analysis, including 16,505 individuals across 21 studies. Region of interest observed in the spatial meta-analysis results, and select regions mentioned in narrative reviews that had not previously been quantified were analyzed. We extracted average FA values in groups and calculated the standardized mean difference, or Cohen's d, for each study. Our results demonstrated that obesity was negatively associated with fractional anisotropy in white matter, meaning that white matter organization was worse with obesity and that water diffusion is less directional or isotropic. The effect sizes for these associations were small to medium in the genu and splenium of the corpus callosum, the middle cerebellar peduncles, and these were all from the spatial meta-analysis. And then from the narrative reviews, we also looked at the superior longitudinal fasciculus, which had a negative association as well. In conclusion, although there are widespread brain changes in gray matter and obesity, the white matter changes that we see are more localized. And although the changes were smaller, the results were highly replicated among both meta-analyses in the genu, the splenium of the corpus callosum, and the middle cerebellar peduncles. We need to think about the implications to cognitive function from these brain changes as well. Again, I'm Lorielle Dietze, and this research was in collaboration with Dr. Hayek's lab and Dr. Joachim Radua. Thank you for listening, and feel free to contact me if you have any questions. Thank you.